மேம் யூடியூப் வீடியோ வந்து ஸ்ட்ரீமிங் ஸ்டார்ட் ஆயிடுச்சு.
வீடியோஸ் ப்ளே பண்ணுங்க ராம். வீடியோஸ்.
Please play the MSc video alone. Please play the MSc video alone. Thank you, Ram. Dr. Stanley has joined us. Kumar, could you please make Dr. Stanley the co-host of the meeting? Ram Kumar, please make Dr. Stanley the co-host of the meeting.
Yes, ma'am. Yes, ma'am. 2 minutes. Yeah, ma'am.
Good afternoon and welcome back, everybody, for the second session of the Ocean Research Knowledge Series 2026, organized by Center for Ocean Research and National Facility for Coastal and Marine Research. We are hosting this Ocean Research Knowledge Series for the 20 years of COR. So we've been in business since 2007, and we are proud to host the 20-year celebration of COR. Today, we'll be having Dr. Stanley Abraham joining with us. Dr. Stanley Abraham is the professor at Center for Ocean Research, and he will be giving a talk today on bioprospecting of seaweeds. Over to you, sir.
Thank you, Ma. Thank you for your kind words.
So you can find it at the bottom, sir. Yeah.
So is it moving?
Yes, sir. It's visible. Moving, sir. Good to go.
So thank you. So today is the first day of 30 hours ocean lecture series, knowledge series. So my talk is on bioprospective of seaweeds. So I am from Center for Ocean Research. So this particular center is being incepted in 2007. So in association with the National Institute of Ocean Technology, Chennai. So earlier in the year 2015, we have inaugurated one particular for 44,000 square feet area in our campus, Satyabama Institute, inaugurated by Dr. Sailesh Naik, Secretary, Ministry of Earth Sciences, Government of India, so which comprises of almost all aquaculture facility, macroalgal facility, and marine bioresources utilization based TBI technology business incubator, which is also again funded by DST, NSTDB, then marine biotechnology lab, microbiology lab, ecotoxicology lab, spectroscopy lab, then microscopy, where we have a very good sophisticated automated systems. And finally, we have genomics and proteomics, fermentation lab, post-harvest, and materials chemistry lab. So apart from this, we had a very good field experimentation facility nearby, near Chamanjeri, that is JPR Salt Pan, then JPR Fishing Harbor at Muttam, Kanyakumari. Then after our continuous credit, we have been sanctioned with Earth Science Technology Cell by MOES. They have given one more facility, national tag, that is National Facility for Coastal and Marine Research, where we have a very good advanced, sophisticated facility. We can't see under one roof in this particular region of southern region. So all the facilities which are being available within our campus under one roof, that is Center for Ocean Research of Satyabama Research Park, Satyabama Institute of Science and Technology. Apart from teaching and training, we are already given one particular program called DBT, Post-Harvest Technology PG Diploma course. So it was run during 2019, 18, 19, 20, 21. So we had a very good skill development techniques for the students who have completed their UG degree with fellowship. And recently we have launched one particular course called MSc Marine Biology course. So we are planning to start from this academic year, and the classes will start from August 2026. So my overview of today's presentation is how we are aligned with sustainable development goals in the particular ecosystem of marine ecosystem. Then my area specialization, one of the area specialization, I am from my microbiology background. So apart from marine microorganisms, I also work on seaweeds. So since 2000, I have been acquainted with seaweeds, cultivation, its product development, and its validation. So earlier we have worked on macromolecules. They are nothing but the first generation of bioactive compounds, whereas the micromolecules or bioactive compounds are known as second generation of molecules that have been extracted from seaweeds, and product have been well developed in global as well as in Indian context. So these are some of the today's topic I'm going to cover. So first you will see SDG. You all know that United Nations has given some target in all these goals to make the resources to be sustainable, and it has to be transferred to the next generation as such. So we are the generation where we are enjoying at most with both technology as well as this has to be uh, along with the nature. This nature has to be given as such to the next generation. That's why they have framed almost 17 goals. And in this, among these 17 goals, we are comes under goal number 14, life below water. So where we have been selected as 86th position 2 years back, and we are topmost in India. So that is very interesting thing, and fortunate to have such a wonderful ranking in this particular goal number 14, that is life below water. So we all know that India has a very long coastal line. So starting from Gujarat to Bengal, we have 11,200 kilometers. So earlier it was mentioned 7,500 after very meticulously planned survey. This Ministry of Ports has given about 11,200 kilometers with an exclusive economic zone of 2.02 million square kilometers. So it almost contains almost all sorts of resources from west to east. So majorly we have been worked in certain aspects related to marine biology. So I'll just give a small introduction about what we did in marine biology. That is open sea cage culture in association with RGCA and CMFRA. We have carried out with the help of deploying a large circular cage of 9-meter width and 12-meter width. So in that we have been deployed these many seeds to harvest and to empower the rural fisherwomen and fisher folk for their livelihood development. So this has been practiced in Olakuda and in Muttam, Kanyakumari. Then one more aspect we have worked in. So earlier it was a finfish. Now it is shellfish, where the craft patterning are being transferred to the local fisherwomen. So this has been particularly trained in this region like Puliket, Alhambra Fort and Muttukad region of Chennai. So where village women have been trained after the tsunami affected area. So they have been given certain kind of livelihood development through this craft patterning. And this has been deployed in JPR salt pan, and it has been well demonstrated in this with the help of mud crab, Silacerata species. Then we are also given marine ornamental fish training to the women for their livelihood development from the region of Kovalam and Chemmanjeri. Then we have also have from the JPR salt pan, we are able to collect the Artemia. Artemia is nothing but they are known as sea monkey. So which has a very good pupa and mufa, that is polyunsaturated fatty acids and monounsaturated fatty acids, which are essential for hatcheries and other aquaculture industries. So from this, we have collected and we have frozen it, and we have given as a one particular economically, that is a viable product, that is for ornamental finfish and as well as for shellfish culture. Uh, they are nothing but they enriched with effective microorganisms. So when this has been incorporated along with the effective microorganisms, it gives a very good gut microbiota for the hatcheries and other animals in aqua industries. So this has been applied in the India Innovation Growth Program in 2014 and under a DST Lockheed Martin program, and it has been selected as 32nd position among 1,400 total technologies developed in India. And apart from that, whatever the fish-based or trash fish has been generated through catchment, through large catchment, they will be let into the environment, or it has been taken to the poultry for developing into a feed. So what we did, we have just developed a certain with the help of effective microorganisms, where we have created and generated, produced trash fish organic fertilizer, which are rich in amino acids and other proteinaceous nitrogenous material, which provides almost nitrogen and phosphorus for the growth of the plants. This has also been funded by funding agency, a national funding agency. So these are some of the area where we have developed our products and it has been marketed. Certain products are well marketed and certain products are being in production when there is a demand in the society. So first we will see about the other coastal and marine biodiversity of India before going into the macroalgal content, that is seaweeds. So we have a rich bioresources. Almost India has 41,000 species of plant and more than 50,000 species of fauna are there. So when it comes to the marine diversity, we have a very good marine invertebrate species, which is the largest among all the species. Then next is the finfish, then marine algae, phytoplankton, zooplankton. Like this, it goes up to seagrass. So we all know that coastal ecosystem provides a wide and vibrant, diversified ecosystem, starting from coastal, then mangrove, seagrass ecosystem, then it goes to the benthic, and finally deep ocean. So like this, there are lots and lots of scope in our marine ecosystems. Okay. So seaweed, we will now come to about the seaweed because it comes under algal group, and they are known as the macroalgae because they are visible in eye when compared to the other microalgal. We have both microalgal as well as macroalgal species. So seaweeds are nothing but they are the multicellular marine algae, so that live near to the shallow region of coastal line, as well as it fix and adhere to the any substrate, solid substrate, like shell. Even shell also it act as a substrate for its growth. Then it's a rocky shore where we can see lots and lots of species are there. In India, the highest diversification of seaweeds are found in Gulf of Mannar, that is almost 300 species have been reported so far. Then when compared to in the western region, Gulf of Kutch, where we have 202 species of seaweeds are being found. So next followed by is the Ratnagiri of Mumbai coast, then Lakshadweep and other regions have its own diversified seaweeds. Okay, so as you said, it will be found in shallow seawater, brackish water, and as well as rocky shores. Initially, it was harvested for its food, cosmetics, and pharma. We have developed one particular unique pulverizer. We have designed our own indigenous pulverizer, which are decentralized one. We can move to the field and it can be driven by the tractor, so that it can be used by at any locality or any region by the fisher folk. So this seaweed fertilizer has been earlier, it was proven and disseminated by CSMCRI. Even that cultivation method is being developed initially by CSMCRI, that is Central Salt Marine Chemicals Research Institute of Bhavnagar, Gujarat. They have developed and they have disseminated, and particularly in Gulf of Mannar, that is from Park Bay to the Tondi, they have a very good self-help model where there are almost 1,000 families are being involved. So initially this was developed and transferred to PepsiCo, and PepsiCo has been transferred to the AquaAgri. And AquaAgri is the only company it's a leading company in India where this cultivation and the product development are being continuously practiced. So we have developed that sap extractor. It has been patented, and we have obtained its patents in 2019. Sorry. Then since it has rich in macromolecules and micronutrients, almost nitrogen, phosphorus, potassium, then iron, then selenium, then copper, zinc, like this, boron. There are so many compounds are being been packed with the seaweed biomass, and this has been utilized for extraction. It's sap containing rich seaweed fertilizer. So apart from that, one interesting and unique feature is the plant growth regulators. There are so many plant growth regulators are being present in the seaweeds as a precursors. That is auxins, gibberellins, and cytokines, which are necessary for its root formation, growth, and flowering, and as well as fruit formation. So these are some of the growth hormones naturally present in the seaweed fertilizer sap. Okay. So I told that I have started my seaweed career since 2000. Initially, we have integrated this particular seaweed cultivation as an integrated farming along with in aquaculture, where it scavenges the nitrogen, nitrates, phosphate, and sulfate as a monoline culture. It acts as a biofilter. It removes the toxic ammonia and as well as sulfur content from the hatcheries. So this has been utilized and it has been well supported by DBT during that time. So myself and Dr. Kameshwaran sir, he has involved in developing the technique for deploying in the as an integrated farming in aquaculture farms. So there is a growing demand. So almost in all the spheres of globe, there is an increasing demand. So still there is a very good potential in seaweed industry. In India particularly, there is a great demand up to 11 lakh tons, but we are producing only 74,000 tons of seaweed biomass. So still the Indian government is giving awareness to go for its cultivation and product development from the seaweeds. Okay. So almost in all the field of application, it is being incorporated. That is food, animal feed, and cosmetics are some of the area which are being predominantly employed using this seaweeds. So we could see almost all the regions are being well utilized for seaweed cultivation in each state of our both east and west coast. So the most interesting thing is the hotspots are being found, that is red highlighted region are the hotspots for cultivation of seaweeds, where the water will be continuously replenished with fresh water as well as replenished with the fresh sea water. Then it is be very shallow so that everyone can enter into the open sea and they can go for seaweed farming. That is one advantage in finding out a suitable spot. So one particular spot is being found in Kutch, Gulf of Kutch, then in Gujarat coast near to Maharashtra, then Ratnagiri and Vizhinjam, Kerala. Then in almost in all southern districts of Tamil Nadu, we can have a very good farming sites. Then in Odisha near Chilka Lake, it is having a very good opportunity for cultivating this particular seaweed. So we will see how it seaweed being differentiated from a terrestrial one. So in terrestrial plants, they have a root, shoot, and flowering parts. Whereas in seaweed, we have a hold fast like a root-like structure. Then a shoot system with a stipe. It will be a large or succulent, or it's a flattened like that. There will be some blade-like structure that is stipe. Then it has some gas bladder, which helps in the for the floating of the seaweed biomass at the surface of the water. So there are three types based on its pigment production. That is those seaweeds which are producing green pigment, they are called as chlorophyceae. Then red pigment, rhodophyceae. Then brown pigment, they are known as pyophyceae. So most of the economically important species are found in brown as well as red species of seaweeds. So these are some of the seaweeds which we are continuously working, starting from sargassum, Padina, gracile area, Apophycus, and so on. So there are different methods of seaweed cultivation because when we are continuously harvesting from the wild, we may definitely one period of time we are going to extinct our very good seaweed resources. So for that we have to culture it and we have to harvest for its economical value added products. Okay, so there are different methods are there. One is the monoline method, then raft method, and third one is the tube net method. Finally, basket method. So there are so many methods are there. This is tube net method. So we'll be having a 25-meter length or 10-meter of length of polypropylene ropes. So in which seaweeds will be incorporated with the help of a tube-like structure of net. So net also should be in polypropylene so that we can avoid any leaching of our microplastics. So this will be added into the tube-like structure. It will be tied along with the mooring structure of anchor or anchor stone so that we have a very good exposed region of the biomass in the surface of the so that we can have a very good growth of a biomass and it can be harvested at particular period from 35 days to 65 days of its culture. Okay, next is the monoline where we'll be having poles at two ends of distance of 25 to 10 meters depends upon our area of cultivation. Then with the help of a long rope, we can insert each inoculum of young seedlings. That is a small thing. So where it can be inserted at an interval of 2 feet gap so that it can grow abundantly and we can get a very good harvest of the. So these things can be cultured in the where the sea is very rough than the shallow region. So this will be suitable for culturing in rough sea. So one particular problem we are facing is the whenever there is a deployed and it has a very good growth of seaweeds, immediately animals will start grazing on it so that it will bite the young ones. That is a tip of the growing seaweed and it causes several diseases to the seaweeds. One particular disease we are facing is the ice disease. So this particular aspect will be covered by one of our speaker in the forthcoming days. So these are the method which are employed with the help of a single monoline rope-like system of culturing seaweeds. Then bamboo raft, it is nothing but a structure of 6 feet to 10 feet length. So it will be like a square-like structure. On each side, it will be having a bamboo or HTP or any PVC kind of material. Whenever you are using in eco-sensitive region, you should not use PVC and HTP so that the material may leach into the environment and cause detrimental effect to the flora and fauna of the marine ecosystem. So always you prefer go for natural raft like bamboo, casuarina, like that. There are so many natural raft are there. So you procure that and make it as a frame. So at the diagonally at the end of the each side, we have to place one small thing, and we have to tie the monoline layer of seaweed inoculants into the as mentioned as shown here. Then for protecting it from grazing animals, it should be covered with a net of size 0.5 inches of the that is 1 centimeter force wise of the net has to be affixed at the beneath so that it can be protected from the any grazing animals or any other poaching organisms. So this bamboo raft can be cultured only in region like shallow regions like Pulicat Lake, then Gulf of Mannar, then certain bay-like structured where we have a very good, very less roughness, sea roughness. Okay, so these are some of the region where we can have a very good system of bamboo raft system for culturing seaweeds. Okay, so this is the raft which you have been trained, given training for the fisher women in Muttam. So this individual rope containing seaweeds will be tied to the each end of the sides of the bamboo raft. Then unique that is 0.5 inches of pore size containing that net will be tied at the beneath of the thing. Okay? And this will be finally deployed in the open sea, and it will be led for its culturing for almost 45 days to 65 days, depending upon the species of the. So in case of Kappa ficus, that is red seaweed, it grows within 35 to 40 days. Whereas for other species, it takes 65 to 75 days, depending upon the seaweed species. Okay, so these are the fishermen. They are involved after during deployment and after culturing the system. Then these are the harvested seaweeds, and these are the monoline method, where between two poles it has been tied, it has been cultured. So these are some of the fisherwomen. They are involved in Rameshwaram region, and these are the seaweeds which are being harvested, and it has been dried in sun dry. And this is one region where we have intensively practiced earlier in 2000, since 2009. That is Muttam Kanyakumari region, where we have a very good breakwall of almost 2.5 kilometer length harbor is there, fishing harbor. So within that region, we are easy to, because Arabian Sea and there is a confluence of both Arabian Sea and Bay of Bengal. So sea is very, very rough. So in that region, since we have a very good closed structure of breakwall, so we have been deployed this raft method of seaweed cultivation in this region. So almost 1,250 rafts were involved initially. Then 620 women were trained. Among them, 127 women are continuously practicing. Then we also disseminated this particular cultivation technology to Paniyur village nearby Chennai. Then Puliket in Tiruvallur district in 2015. Then recently with the DST seed program, we have been continuously training certain science and technology intervention for tribal community. Okay? So we are making them as a empowerment in seaweed farming and to develop and extract and product development on their own. So almost 160 members have been trained so far. Sorry, 240 members have been trained so far in 3 regions. One is in Viluppuram district, Talangadu Marakanam block, and this was again in the Markanam block, and this is in Alangankuppam, Kadapakam in Chengalpattu district. So these are the beneficiaries. Beneficiaries. They are getting trained for in tube net method, because tube method is very, very fast growing and well protected from grazing animals. So we have a very good possible of its thing. Since we are being deployed in backwater, in Puliket we have a very good growth, and in Talangadu we also very good growth. Next is the Kadapakam region, where it has a very less growth. So like this, area of selection also very, very important. Okay. So area of selection is how we are going to select is it should have minimum 3 feet depth. Then it should be free of any pollutant, that is particularly from domestic sewage or any form of pollutant should not affect its growth. So that is very, very important. Then after selection of the thing, we have to fix it a particular region where we can easily go and collect and come. So these backwaters are very easily accessible by the fisher women and fisher folk. So we have selected these regions like Talangadu in Markanam, Kadapakam in Chengalpattu, and Puliket in Tiruvallur districts. So where the tribal community has been given training and they have been involved in deploying these seaweeds. And after culturing, we ourselves procure for its commercialization. So this is in Minjur block, Puliket region. Okay, so after culturing and after harvesting, so there is a very good value-added products are being developed or extracted from these seaweed biomass. Okay. So we all know that phycocolloids are nothing but stabilizing agent. Then food stabilizers, it has been well utilized. So there are 3 major phycocolloid are being exploited for its commercialization, that is agar, alginate, and carrageenan. So these are extracted by a very good hot alkali method, which has been developed by Food and Agriculture Organization. So where we can harvest the phycocolloid and it has been refined and it can be directly marketed. So when we are purchasing the seaweed, it will cost only selling the seaweed to fetch you around 25 to 80 rupees per kg. Whereas if you have been processed, it will be fetch you around 1,000 depending upon the quality of the phycocolloids. So these are the methods which are being developed and it has been given by a Food Agriculture Organization, FAO. So this is the simple method for extraction of carrageenan with treatment of potassium hydroxide and sodium bicarbonate. And this alginate particular thing, it need a 1 day process. So it is also followed by the hot alkali method. So after culturing in these regions, we can check its yield of the phycocolloid, gelling temperature, melting temperature, and gel strength. So gel strength is very, very important for its commercialization. Then its purity also is important. So whatever we have harvested, it has a very good yield when compared to the other region. So that one interesting thing we have found during our culturing of the seaweeds. So these are the economic cost benefit analysis when we are directly culturing with the help of both raft and as well as monoline system. So we have up to 400 kg of more growth will be obtained from almost 25 to 50 ropes in monoline culture, and we can extract around 60 to 75 liter of thing, so which it can fetch you almost 7,000 to 10,000 liter, 10,000 rupees for its one cycle of harvest. So this will be given, this economics will be given for one particular family or one particular member involved in seaweed farming. So whereas if you are taken as a biomass has been converted into a phycocolloid extraction, it gives a very good in return. So whereas you can get almost 5,000 to 6,000 rupees of per cycle. So that is from one particular cycle for one particular row or raft. This is the cost benefit analysis we have been achieved so far. Then apart from that, since these SAP has high macromolecules and micromolecules, so we have a very good nitrogen content, phosphorus and potassium. So since potassium is high, this can be replaced for any form of potassium based fertilizer in Indian context, because we are importing certain potassium fertilizers. So this will be an alternate for its generally available market available potassium fertilizers. So this is the in a nutshell. So these are the extractors which we have been designed, we have been fabricated and applied for patent, and we have been disseminated to almost more than 12 villages in Tamil Nadu for its value-added product extraction. And this is the highlight of our seaweed liquid fertilizer. It is used for almost for all crops. Then there is an increase, almost about 40% increase in the sugarcane, whereas from in other cereals and pulses and vegetables, there are about from 15 to 25% are being increased in it. Okay. So recently we have also given for this cut flower industries in Hosur, where they have a very good yield when compared to the previously used commercially available foliar spray of fertilizers. So next we are moving to the second generation of seaweed bioactive molecules. So earlier the phycocolloids are known as first generation of molecules, whereas the second generation of molecules bioactive molecules are novel compounds that are being extracted, and it is still in certain molecules are in research, whereas in several institutes like CM4I and even of course our Center for Ocean Research, then NIFTM, they are continuously extracting all the bioactive molecules from those seaweeds, and it has been well established for its applications, for its diversified applications. So they are nothing but polyphenolic compounds, alkaloids, terpenoids, flavonoids, and vitamins, pigments, and other secondary metabolites. So these are the major second generation of compounds. So these aspects has to be concentrated for its high market value. So in future, definitely it's going to happen. So we have worked in a certain carbohydrates. So these are the results which shows our research on seaweeds in Center for Ocean Research. So we have been extracted the polysaccharides like fucoidan, agar, alginate, carrageenan, laminaran for its anti-diabetic, antioxidant, anticoagulant, anti-cancer, anti-aging, as well as immunomodulatory. So this immunomodulatory is very useful during COVID time because during that time we are trying to develop certain immunomodulatory effect of interleukins. So it has have a very good immunomodulation has been happened during that time. So we have also incorporated this particular thing for our easy cure of our during pandemic time. Okay. So apart from that, we have also studied about in carrageenan and farfirine and ulvan of chlorophyceae. So we are also extracted certain oligosaccharides and monosaccharides from these polysaccharides. So the like fucoid and carrageenan are well-known source of antioxidant and coagulation, anticoagulation and immunomodulation. So apart from that, we have also studied about its mitochondrial reactive oxygen species, which reduced with the help of the glucuronate. Okay. So what are the sulfated polysaccharide that has been extracted from brown type of seaweeds, sargassum and padina? So we get a very good source of sulfated polysaccharides from sargassum tenera. Then these are some of the individual sugars like fructose, galactose are the predominant sugars in the sulfated polysaccharide. So, and this has been evaluated for its monosaccharide content, molecular weight, and other aspects of bioactivity, which are extracted from Padina gymnospora. Earlier, it was Sargassum tenerifum. Now, we have also studied evaluated potential in gymnospora, Padina gymnospora. Then we also evaluated its anti-cancer activity, that is apoptotic potential of these sulfated polysaccharide. So when compared induced with the hydrogen peroxide, the more cells are being live when we are being substituted with sulfated polysaccharide in increasing concentration from 25, 50, 75 and 100 microgram per lemon. So we have also studied for its anti-aging potential, that is senescence associated beta galactosidase assay. So where it has a very good anti-aging effect when this sulfated polysaccharide particularly used for its hydrogen peroxide induced. Then we are also interesting thing we have found that the secondary metabolites like phenolic compounds, flavonoids are rich in during new moon than the full moon. So this particular thing which helps the researchers to collect the seaweed species particularly during new moon day. So and we have also analyzed for its what type of compound are being there. So phenolic compounds are very rich when compared to the other compounds during new moon day. Okay. Then we have gone for extraction of carotenoids. One particular carotenoid we have extracted is the ficloxanthin from Padina. So this is the characterization of those things along with the control. Standard has been utilized, used here for comparison. The method we have employed is the solvent extraction method, then ultrasound assisted method against a microwave digestion method. So ultrasonication gives a very high yield when compared to the other two methods. This has been employed for extraction of particular ficloxanthin from Padina species, and it has been well established against, studied and evaluated against its anti-aging. So like this, these are the compounds. Apart from that, we have studied its antibacterial, anti-cancer, antiviral activity. So antiviral activity we have evaluated. So which is high when this particular species is being used. That was polysaccharides that have been employed for. Okay. So when there was some potential of generating its nano composites. So we have used certain seaweed extracts for development of nano composite material, so that this nano composite material we have employed for catalytic converters. So you all know that catalytic converters, conventionally available catalytic converters, they are utilizing this platinum as one of the catalysts, whereas here we have used nanomaterials that are being generated using seaweed extracts. So they have effectively detoxify the uh, carbon dioxide, carbon monoxide, and as well as hydrocarbons, which have been emitted from the exhaust of the two-wheelers, vehicles, automobiles. So this is the structure of that air detoxifier. So which it has utilizes the nano composite generated from the seaweed. So apart from this, there are so many edible type of seaweed compounds are being there. So in different forms, nowadays in India also we could see in several restaurants they are providing salads and soups which are made using stick. In India, we can see there's a China grass which is being imported from, because we are harvesting and exporting the biomass, and they are value added giving the and providing as a agar. So this has been available in all supermarkets, and it has been well utilized for its custard and other jelly like thing for development of the chocolates, then other desserts. Okay. So these are some of the compounds which are available in the market. Then apart from that, we have cookies are there, jellies are there, then jams and marmalades are there. So then certain food spreads are also there. So these are some of the commercially available seaweed based products in our Indian context. So the CM4I, they have developed a very good composition of products along with the other seafoods, so that to increase its value and as well as longevity of the product. Then in IFTAM, they have developed, this is a MOFI Institute, Ministry of Food Processing Institute. So there they have developed one particular tea which has been infused with the seaweed extract. So they are rich in polyphenols, since they are having more phenolic compounds. So these polyphenols are act as a very good antioxidant activity and other anti-cancer potential of the. So this has been evaluated for its thing, and the final validation is on process, so that we may have the product within some 1 or 2 years. So there is a growing demand for generally for seafood, particularly for seaweeds. So there are increasing utilization of the seaweed-based products in our market and as well as in a culinary purpose. So apart from this culinary general foodstuffs, we can also incorporate it in cosmetics and other beauty products. So where this nutraceutical potential has been well utilized, and given for hyaluronic acid from certain species and squalene molecules from certain species, they are being validated for its nutraceutical property, which are in progress. Okay, so these are the some of the highlight of my presentation. So we always go for vocal for local. Then what are the products we are developed through our research? It should be reach the market, or it should benefit the society through its commercialization for Atmanirbhar. So I would like to conclude. So with this, so there is a very good demand for all sorts of sea, seafood products and sea-based value-added products from our. So if you have any further questions, I'm here to answer, or you can mail or you can chat through me with this WhatsApp number. Okay. So any more questions, or over to Samya madam. Thank you.
Thank you, Dr. Stanley, sir, for your very elaborate talk on seaweeds and their applications in marine biology and biotechnology. With this, we have now come to the end of the second session on the Ocean Research Knowledge Series. Participants, if you have any further queries or if you wish to reach out to us, please find the link for our official website on the chat box. So you can reach out to our research experts through the contacts provided in the official website. Thank you, sir. And let me just share my screen for a while. Is my screen visible?
Yes, ma'am.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
So tomorrow's lecture series, the expert lecture on seagrasses, a hidden forest of the sea in an era of climate change, will be handled by Dr. D. Jayaprakash, Assistant Professor Research, Center for Ocean Research. This will be scheduled at 10 a.m. tomorrow. And the next talk by Dr. M. Ravi will be on polychaete taxonomy and DNA barcoding, which is scheduled for 2 p.m. to 3 p.m. So looking forward to join you all tomorrow at 10 a.m. and at 2 p.m. Thank you. Thank you, sir.
Thank you, ma'am.
And thanks to all the participants for joining us today. We wish your continued support and your presence tomorrow for the